Techniques to study secondary metabolites. What are secondary metabolites? Secondary metabolites are organic compounds produced by organisms that are not directly involved in the growth, development, or reproduction of the organism. Unlike primary metabolites, which are essential for basic life processes such as metabolism and growth, secondary metabolites are often considered non-essential but play crucial roles in various ecological and physiological functions. Plant secondary metabolites are categorized into terpenes, phenolics, and nitrogen-containing compounds. Next are the techniques to study secondary metabolites. Studying secondary metabolites involves a variety of techniques given their diverse chemical structures and biological activities. Here are some commonly used techniques. First one is extraction. Secondary metabolites are often present in low concentrations in organisms. Extraction methods involve using appropriate solvents to isolate these compounds from the biological matrix. Common extraction techniques include oxalate extraction, maceration, sonication, and liquid-liquid extraction. And the next one is chromatography. There are several types of chromatography, and the first one is thin layer chromatography, which is used for preliminary separation and identification of secondary metabolites based on their different affinities for the stationary phase. And the next one is column chromatography. This technique is used for larger scale separation of secondary metabolites. They are separated based on their differential partitioning between a stationary phase and a mobile phase. And the next one is high performance liquid chromatography, which offers a higher resolution and sensitivity compared to column chromatography, making it suitable for quantitative analysis of secondary metabolites. And the last one is gas chromatography, which is used for volatile secondary metabolites, which involves the separation of compounds based on their vapor pressure and affinity for the stationary phase. And the next technique is spectroscopic techniques. The first one is mass spectrometry. Mass spectrometry, it is used for identifying the molecular weight and structural elucidation of secondary metabolites. Techniques such as LCMS and GCMS are commonly used. And the next one is nuclear magnetic resonance spectroscopy, which provides information about the structural arrangement of atoms within a molecule, aiding in the elucidation of complex structures of secondary metabolites. And the next one is infrared spectroscopy, which is used to identify functional groups present in secondary metabolites, aiding in structural characterization. And the next are bioassays. These are used to evaluate the biological activities of secondary metabolites. They include assays for antimicrobial, anti-cancer, antioxidant, and other pharmacological activities. And the next is genetic techniques. With advances in molecular biology, genetic techniques such as gene cloning, expression, and manipulation are used to understand the biosynthetic pathways of secondary metabolites. This include techniques like PCR, gene knockout, and heterologous expression. And the next is bioinformatics. Computational tools are used to predict biosynthetic gene clusters and pathways involved in the production of secondary metabolites. This helps in understanding the biosynthesis and regulation of these compounds. And the next is metabolomics. This involves a comprehensive analysis of all metabolites present in the biological sample. Techniques such as LCMS, that is liquid chromatography mass spectrometry, and GCMS, that is gas chromatography mass spectrometry, are commonly used in metabolomic studies to profile secondary metabolites in complex biological samples. And the next is chemical synthesis. Total or partial synthesis of secondary metabolites can be performed to confirm their structures and investigate their biological activities. By employing these techniques either individually or in combination, researchers can effectively study secondary metabolites, elucidate their structures, understand their biosynthesis, and explore their biological activities and potential applications. Thank you.